Hello everyone. Today I will share a fascinating breakthrough in structural biology, specifically regarding the molecules that hold the blueprint of our life, RNA. We will review one of the Nature article published on July this year, titled Naturally Ornate RNA-only Complexes Revealed by Cryo-Electron Microscopy. For years, there were a huge gap at the field of large non-coding RNAs or ncRNAs. Out of over 4,000 known cases, only about 140 had their 3D structure solved. The prevailing idea was that if an RNA molecule was large and complex, it absolutely needed a protein or a small molecule partner, a kind of scaffold, to help it fold and remain stable. But our three candidate molecules, bacterial RNAs named OLE, ROOL, and GOLLD, which I will refer to old rule gold for convenience, told a completely different story. They are known for these elaborate, ornate secondary structures, but how they folded up in the cell was a total mystery. Now, here's the key finding. Using high-resolution cryo-EM, they have determined the structure of all three, and they are truly interesting. Unexpectedly, they form highly ordered, symmetric complexes without any protein involvement. These RNAs are folding themselves into complex three-dimensional homo-oligomers. To prove this wasn't just a high-concentration lab artifact, they performed robust biophysical tests using mass photometry and dynamic light scattering. The results were conclusive. The distinct stoichiometries, a dimer, an octamer, and a tetradecamer, are stable at nanomolar concentrations, which is well within a biological range. This confirms that these molecules are genially programmed to self-assemble. Now let's look at the structures, starting with all RNA. All forms a compact diameric complex, essentially a bundle of paired A-form helices that they describe as long coaxial pipes. The two chains are held together by intricate intermolecular connections, which they call bridges. One of the most conserved connections is Bridge 1, featuring a symmetric 4A by A base pair interaction. This complex folding mechanism suggests All's role is not just structure, but organizational. All is known to bind proteins, and our structure reveals that its binding sites are already structured and accessible in the RNA dimer. This supports the hypothesis that the all dimer forms first and the RNA itself then serves as a scaffold to organize proteins, like potentially helping OAPA form a functional membrane-associated tetramer. Now let's move to the first of the two nanocages, rule RNA. Rule is a remarkable structure that self-assembles into a large, closed, hollow octameric nanocage. It has deeper symmetry and a diameter of about 280 angstroms, which makes it even physically larger than a bacterial ribosome. The interior of this complex is essentially empty, encapsulating only a disordered linker region from each chain, but the surrounding framework is incredibly stable, held together by multiple kissing loops and A-minor interactions. The largest structure is gold RNA. This molecule forms a gigantic tetradecamer nanocages with D7 symmetry, stretching to 380 angstrom in diameter. Gold offers a structural contrast to rule. Here we see a clear domain separation. The 5' region forms the cap and the 3' region forms the ring. This modular organization is likely what allows the sequence of these two domains to evolve somewhat independently across the homologs. Like rule, its closure is mediated by complex intershell interactions, notably the self-interacting kissing loops. So how does self-assembly happen? It's all in the non-canonical motifs, the structure glue. The stability of three complexes relies heavily on the elements like A minor interactions, wherein adenosine docks into the minor groove of an helix, and the classical kissing loops for loop-by-loop base pairing. Crucially, the biological relevance is backed by evolutionary analysis. When you look at the thousands of homologs, the sequences are at the intermolecular contact sites, the bridges, were highly conserved. This suggests a strong evolutionary pressure to maintain the ability to form these large assemblies. Furthermore, our controls were clear. A non-protein dependent RNA remained disordered, while other large RNA folded only as a monomer, proving the self-assembly of all rule and gold is both intrinsic and specific. This brings us to the big questions. Why build an empty cage? The primary function of the hollow rule and gold nanocages is hypothesized to be molecular encapsulation. They might act like protein axios bacterial microcompartments designed to capture and hold metabolites, stress proteins, or even tRNAs. They might also serve as dynamic containers capable of opening and closing to encapsulate something as large as the ribosomal subunit. In summary, this research is powerful demonstration that RNA is capable of large-scale macromolecular self-organization and encapsulation on its own. It expands our understanding of RNA structural roles far beyond simple regulatory or catalytic functions. The next step they are suggesting is clear. We need to identify the specific cargo inside the rule and gold cages and understand the biological triggers that control the assembly and disassembly. And I think that's it. I hope this presentation was useful and helped you better understand this topic. And if you have any questions, you feel free to ask. I'll be happy to answer them. Thank you.